HMOs are a very diverse class of oligosaccharides. So oligosaccharide basically means many sugars. So these can be as short as 2, like a lactose with fucose or sialic acid on it, to very long sugars. What we've known about HMOs is they seem to have many functions. So they can be prebiotics to healthy bacteria, in particular bifidobacteria. So if you think about the HMOs, they're the fiber of human milk because we do not have the digestive enzymes to break the sugar bonds. So they pass through our gastrointestinal tract to the lower bowel where the bacteria can utilize them. So they can ferment them, they can make components like short-chain fatty acids, which are beneficial to the host, or the bacteria can also use them to grow and proliferate. So that's why they're considered a prebiotic. And the other functions of the HMOs are that they mimic the sugars that are on cell membranes that many pathogens bind to. So certain bacteria and viruses will bind to fucose or sialic acid. And since HMOs have these sugars on them, many of them have these fucose or sialic acid, they can basically block the binding of these pathogens to the infant's gut. And therefore, they help to bind up the pathogens and they are just swept out of the body and they don't actually cause an infection. So those are some of the local benefits in the gut. But emerging data also suggests that they may be benefiting cognitive development as well as systemic immunity. Studies done in cell cultures show that if you put HMOs on these cells, they can impact how those cells proliferate and how they differentiate. And what it seems to do is it stops the cells from proliferating, so growing, basically making new cells, and pushes them to do what we call differentiate, where they begin to look more like mature intestinal cells. So they will start to express enzymes and transporters. So it does seem like some of the functions are to help the gut become, more functional as a gut. Studies in piglets, for example, and that we do in my laboratory or in humans clinical trials, haven't necessarily shown these effects. And it could be that in cell culture, all the cells are very similar to each other, and they're usually all at the same stage of growth, whereas in the gut, cells are at all different stages of growth and development. So it may be more difficult to see these effects. But either way, a number of labs have shown that the HMOs can directly impact cells. The HMOs that each woman makes are unique. And studies have shown that there's actually up to 200 different forms of HMOs, although there's about 15 or 16 that are present in most women, and we kind of think about as a core HMO. So most of the rest of them are in relatively low abundance. One of the main factors that influences HMO composition is what we call the mother's secretor status. And secretor status is also related to blood group antigens. And in this case, it's the gene called FUT2. And this gene encodes a protein called mucosal 1,2 transferase, alpha 1,2 transferase. And this puts 2 linked fucoses onto cell membranes, but also onto lactose when HMOs are made. So one of the major HMOs in the milk of secretor moms is called 2FL. It's just lactose with fucose on it. If that mom is a non-secretor and therefore doesn't have this enzyme, she does not produce any 2FL in her milk. And there's also a couple other HMOs that are longer that have 2 fucose added to them. So what we see in these moms that are non-secretors, the lack of these two linked fucoses and a reduction in the total HMO content of their milk, so that we see less total HMOs. And, is this important? There are some studies that suggest that when moms are non-secretors, that their babies can have actually a higher incidence of diarrheal diseases. These are studies that were done in Mexico. So because many viral pathogens and bacterial pathogens bind to fucose on the cell membranes, the lack of the 2-FL in the milk is not conferring that protection, not inhibiting those pathogens from being able to bind to the baby's intestinal cells. The other thing that's been shown is that the microbiome of the babies whose mom is a secretor versus a non-secretor differs. And for example, the babies from non-secretor moms have lower bifidobacterial abundance. So we know that there are some differences between the moms. Overall, about 30% of women are non-secretors. It differs by country. In the US, it's 25 to 30%. In China, it's about 30%. Whereas, for example, in Mexico and South America, it's very low. It's only like 2%. So as we look globally, the HMOs that moms produce is different, and a lot of it depends on the secretor status of the moms. Summarized a lot of an overview of HMOs. So a little bit of what we've talked about which HMOs are there, what their general functions are, and then I also presented the results of 2 different clinical trials that have been conducted to give the audience a flavor for, what are the potential advantages of having these HMOs in formulas. To date, the 2 studies that have been done, one is just with adding 2-FL, and the other is adding 2-FL and LNNT, have shown that the infants can grow normally, have normal things like their stool consistency, spitting up. So, really no differences in tolerance of the formula. So to date, there doesn't seem to be any adverse effects of having HMOs, and then there are some potential beneficial aspects that I discussed. For example, improved immune function and reduced disease. I presented some of the work that we're doing in my lab looking at cognitive development and what human milk oligosaccharides might be doing in terms of cognitive development. And the reason that we're interested in that is that we know that both fucose and sialic acid are important in the brain. Fucose is important in synapses signaling, so how the brain is communicating. And then sialic acid is also part of gangliosides in the brain. And so these are also very important bioactive components. And studies of infants who, unfortunately had passed away, when they did autopsies, they found that babies who had been breastfed had higher levels of sialic acid in their brains than babies that were formula fed. And we know that there's also links that breastfed babies have higher IQ scores than formula fed infants. And so because of the importance of sialic acid, there's been this hypothesis, this question of whether the HMOs could be contributing to that. So we've done a couple studies. So this is in collaboration with my colleague Ryan Dilger. We've done some human studies, but also piglet studies where we're feeding them different HMOs as well as what we call BMOs. So these are oligosaccharides from bovine milk, which are mainly the sialylated ones. So, what we were able to show is that when we fed the piglets formula with HMO, they had better learning and memory, and that there were also differences in the size in various brain regions. So we're currently continuing those analyses, but it suggests that there may be ways that HMOs are actually impacting the developing brain and improving their ability to learn and remember things. One of the challenges is that human milk is just so complex, and that there's bioactive proteins and lipids and carbohydrates, and they likely are working together in very interesting ways that we don't fully understand. And oftentimes when we're trying to do studies, we will take, for example, an infant formula and just add HMOs, or just add osteopontin that Dr. Lonergan talked about. And that just gives us, it helps us to understand, does this one component exert an activity, or is it bioactive? But it doesn't allow us to fully understand the complexity of human milk. And so that's one of the challenges, because I believe that these components are actually interacting and working together. And so in the future we need to be able to tease out these effects, because a goal is to make formulas as close to human milk as possible. I mean, obviously breast is best, and we want as many women to breastfeed as exclusively and as long as possible. But when that's not possible, then to have the best quality formulas. But we always have to balance adding new ingredients with, are they really efficacious? Are they actually having a benefit to the infant? Because we want to be mindful about the fact that when we add a single ingredient or just a couple ingredients, that we may see benefits, but we may not necessarily know what long-term effects might be.